Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
From salamander tails to deer antlers, the innate ability of some animals to regenerate lost body parts has captivated scientists for centuries and driven research into the molecular mechanisms of such healing in the hopes that humans might someday be able to do the same. Even amongst regenerating organisms, hydras, not the mythical beasts, but aquatic animals in the genus Hydra, stand out for their ability to regrow any part of their body after it's lopped off, including their heads. Now, a study published last week, December 8, in Genome Biology and Evolution sheds new light on the genetic and epigenetic mechanisms that allow these animals to achieve this regenerative feat. Hydra vulgaris are 1 to 3 cm long, tube-shaped, freshwater organisms that adhere to objects such as sticks and rocks and, much like their relatives the sea anemone and jellyfish, hunt using stinging tentacles. They are considered to be immortal. Unless something comes along and kills them, they will continuously replenish lost tissues and body parts. They can also clone themselves through an asexual reproduction process called budding, in which a new hydra grows out of the original and detaches, kind of like the way petals would come out from a flower. University of California, Irvine, cell biologist Ali Mortazavi tells The Scientist. It's a very interesting question because it's one of the mysteries of cnidarians in general. They have all these fantastic modes of reproduction, and the big question is, are they recapitulating some process? Is regeneration like budding? But we don't really know so much, says Chiara Sinigaglia, a developmental and evolutionary biologist at the Institut de Génomique Fonctionnelle de Lyon who didn't work on the study. This mystery is a question that people have been trying to address from different angles, for years, Sinigaglia adds. It's very tricky because you see the same genes used over and over in both processes. For instance, research published in eLife in March highlighted the role of the WNT signaling pathway in rebuilding a Hydra vulgaris's mouth after decapitation. In contrast, Mortazavi and his colleagues took a more comprehensive approach, mapping out which genes were activated and upregulated at different points in time and in various tissues throughout the budding and regeneration processes. The team followed that up with ATAC-sequencing, a technique that identifies open-chromatin regions of a genome, to find out which genes were accessible to those candidate transcription factors. For both reproduction and head regeneration, Hydra vulgaris rely on their head organizer, a cluster of 50 to 100 cells at the apical tip of the head that signals nearby cells to differentiate into either part of the head or the tentacles. When the head is cut off, the researchers found, a cascade of genetic and epigenetic activity rapidly triggers the growth of a new head organizer from whatever cells remain, allowing the Hydra to regenerate the rest of its head as well. When severed, this solitary tunicate regrows as 3 new animals. This paper, it's not giving us the exact answer, but it's generating the raw material, Shrivastava says. I think it's great for hypothesis generation. Emergency reconstruction. As the March eLife paper indicated, a decapitated Hydra will quickly reassemble its mouth as a survival mechanism. In contrast, a budding Hydra clone undergoing normal development doesn't need to prioritize wound healing or survival, he says, and can progress at its own pace. Even though the processes are different, they still use the same developmental factors or genes, just in different ways. Lead study author and University of California, Irvine, evolutionary biologist Aide Macias Munoz tells The Scientist. I think that's because it's easier to co-opt things that are already doing something. They're already being used in development to develop a head, so why not use similar things to develop a head again? But this is a different process because you need to do it very quickly and stop it at some point. First you want to look at it one head at a time, and then you want to look at it with thousands of cells per head. In order to test and confirm the gene's roles, as well as those of the candidate promoters and enhancer regions, researchers will need to conduct functional knockdown or knockout studies, Macias Munoz explains. Some of that work is already underway. Hydra has been part of that story for a long time. Researchers first began studying its regenerative capabilities in 1744, when Abraham Tremblay first discovered the bizarre property. But other organisms have garnered more attention and detailed study, and many questions about Hydra regeneration remain unanswered. Experts still don't know whether regeneration is a shared ancestral feature that emerged in some ancient organism, or whether creatures such as Hydra, the worms studied by Shrivastava and Benham Pyle, mammals, salamanders, and others that can regenerate some or all of their lost body parts developed. The paper notes that some of the tools necessary for regeneration must have emerged before the phyla to which Hydra belongs, Cnidaria, split off from the rest of the animal kingdom. One likely possibility, Sinigaglia adds, is that both regenerating and non-regenerating organisms inherited a broadly similar toolkit. WNT3 is expressed during embryonic development even in non-regenerating species, for example, which regenerating organisms then independently adapted to their own brands. It's kind of nice that we're seeing similarities across all these different organisms, says Benham Pyle, because it makes it more likely that we might be able to leverage that biology to help humans. Quote, "Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you."